All living organisms are comprised of cells. They can be single-cell organisms like algae or complex multicellular organisms like humans. Within the cell there are many different organelles, all crucial for cell replication. The initial line of defense of a cell is the membrane. The membrane regulates what is able to come in and out of the cell. It is also an avenue for cell-to-cell communication. In a normal cell, the cell cycle regulates cell proliferation by regulatory proteins or enzymes. These regulatory proteins/enzymes provide the checkpoints to move forward to mitosis, proceeding to cell replication.
The blueprints to make a new cell are in the DNA, wrapped up inside 23 pairs of chromosomes. At the ends of the chromosomes are telomeres. The telomeres act as a protective cap to the ends of the chromosomes. Another way to think of a telomere is like an aglet, the plastic cap on the end of a shoelace. In the process of cell division and aging, the end pieces of chromosomes can become damaged or fall off. When the telomeres can no longer protect the chromosomes, DNA mutations occur. When the cell mutates, multiple mutations can cause the cell to become cancerous.
In a cancer cell, the regulatory processes are overridden, allowing for uncontrolled cell proliferation. This is achieved when a cancer cell creates their own hormones or receptor sites to induce replication. These imposter proteins push past the checkpoints in the cell cycle, also avoiding apoptosis, programmed cell death. This causes rapid cell growth, which then creates tumors, leading to metastasis. Metastasis, from the Greek change of place, is the movement of tumor cells from the site where the cancer began to grow in other sites of the body. It does this via the lymph system, through the gaps in the lymph vessels.
Cancer cells mutate at a rapid rate. They proliferate due to the fact that the checkpoints are bypassed and they don't recognize the programming data of the cell. Cancer can grow at a rapid rate through communication of secretion of fluids and the development of receptor proteins on the cancer cell membrane, as opposed to non-cancerous cells that have antigens triggering cell death.
We looked at a possible solution to cancer by implementing two safeguards: regulating the proteins or enzymes within the cell cycle and protecting DNA. The proteins/enzymes in the cycle can be manipulated in order to stop the cell from advancing through the cycle to create new mutated cells. Telomeres can help to prevent a chromosome from becoming damaged, since they are the end caps to DNA. If we use the enzyme telomerase reverse transcriptase, triggering the telomeres to grow, then the DNA will be more protected from becoming damaged or mutated during cell division. Our design for the perfect cell is based around these concepts in the cell.